So our talk is going to be held by Sid, who's currently an undergrad at UC Santa Barbara. He's also been a longtime member of the Vegan Cheese Project, which is hosted by BioCurious and Counterculture Labs. And he has been doing some of the bioinformatics for our Narwhal weird offspring of the Vegan Cheese Project. So I'm gonna hand it over to Sid and take it from there.
All right, thank you, Patrick. Let me just present this really quickly. Let's see, how's that? Is that working for now? I think it should. Perfect. All right, everyone, welcome to On Narwhal Cheese, the Evolution of Whales and the Imaginary Phylogeny of Unicorns. So before we get started, I'm gonna have a couple demos in this workshop, and the easiest way to get to all those links is through this links document. I'm going to be posting it in the chat right now as well. Let me see if I can get there. There we are. So that's the links document right there. And then the next thing we want to tell all of you is we'd love to have all of you at our weekly meetings. It's Monday, 7 to 9 p.m. Pacific time. And that link should also have been posted in the chat as well. So as an outline of what we're going to be going over today, so we're first going to overview both of the Real Vegan Cheese Project and then the spin-off Narwhal Phylogenetics Project. And inside of the Narwhal Phylogenetics Project portion of the presentation, I'm going to be doing a quick demonstration on how we search for annotated genes, one of the bioinformatics techniques we've been working with. And then we're going to look into two of the studies that we're doing with our Narwhal project, which is the Casein gene cluster evolution study and the TUS development study. And in both of those, we're going to look at the Casein gene cluster with recent data. And then we're going to make some FASTA entries and then align some sequences and make phylogenetic trees out of this. So to introduce our Real Vegan Cheese Project, the whole project idea is to make real cheese through engineered bacteria, right? So in theory, we would take a portion of the cow's genome that codes for the milk proteins and have these bacteria make those proteins for us instead of having the cow make the milk. And then from there, after making those proteins, we'd make our sort of synthetic milk, which will then be used to make cheese. And we would go through the whole cheese making process then. And the end goal is to make cheese that is almost identical to cow's cheese in texture, flavor, and all of its melting properties and everything without the downside effects of using an actual cow, which would be the carbon emissions and animal cruelty. So in theory, right, we're going to be all we need is a little portion of a mammal's genome that codes for these milk proteins. So in theory, we can kind of do some crazy stuff, right? We can start thinking of a bunch of other animals. So moose milk is sort of a delicacy. So with this technology, you can start mass producing that if you wanted to, but this really got us thinking, what's the craziest animal we could make what's the craziest animal we can make milk out of? And that gets us to narwhals, right? The narwhal genome was published recently back in 2019, and we are really looking to study how the casein gene cluster, I'm going to explain that in just a second, evolved in mammals, and then we're going to look at how the tusk developed in the narwhal, which is interestingly somewhat connected to our casein gene cluster. So first off, let's explain what milk is before we get into a bunch of these things. So milk is basically a combination of water, sugars, fats, and proteins at different proportions based on the animal. However, casein is the protein that makes up milk. And there are 4 types: alpha S1, alpha S2, beta, and kappa caseins. And all 4 of these are not found in all mammals. Some mammals have all of them. Some mammals have a small portion of them. However, beta and kappa casein are almost always found in every mammal. Now, this gets us to our first important concept, which is the casein gene cluster. So these are figures from a genomic study done in 2003. And you can see these CSN blocks right here represent casein genes. And if you notice in all the human, mouse, rat, cattle, all these genes are located very close to each other. So instead of individually studying these 4 genes, we sort of group them together and study all 4 of them together, or however many the organism has. So this gets us into our first workshop. So again, we can refer to our links document. It's going to be the first one under searching for annotated genes, and that's going to take us to our website, which is going to be GenBank. We're going to be searching for annotated genes, which is the first step we took in investigating this casein gene cluster. So if I can get some, let me see if I can get the participants list. If I can get some reactions from everyone when you guys have that page open, that would be great. And I'll just give a couple more seconds for that, and then I'll move on. All right. Perfect. So I'll post that links document once again in the chat, and we'll move on. So starting off, when we get to GenBank, we're going to be presented with this website. It's basically, you can think of it as a Google search, right? So the first thing we want to do is make sure we're searching the sort of the correct subdatabase. So by default, it should be searching for nucleotides. If not, just quickly click the drop-down menu, make sure you select the nucleotides, and then we're going to look at the search query, right? And for us, it's going to be human kappa casein. Again, all these search queries and links are in the links document. So once we do that search, we're going to be presented with a couple results. So the one we're going to be looking at is the human kappa casein gene, complete CDS 9844 BP linear DNA. So when we click on that hyperlink, we're going to be presented with a lot of information here. So starting off at the top, we have this 9844 BP of DNA. So this 9844 BP stands for 9844 base pairs or nucleotides. It's the size of this sequence. And then the DNA is just specifying the kind of nucleic acid. So the nucleotide sort of subdatabase can have the mRNAs, the DNAs, and sometimes some miscellaneous RNA sequences in there. Now, the next thing we want to look at is the definition. So this is the human kappa casein gene. And since it is an annotated genome or an annotated gene, we have this information for that, for the sequence. And by annotated, I mean that the sequence has been labeled a purpose. A lot of these genomes have been sequenced, but the specific nucleotide sequences haven't been labeled a specific purpose. And in this case, fortunately, we do have a purpose on this one. Next, we're going to look at the accession and version numbers. If you notice, they're very similar. The version number is basically the same thing as the accession number, but just with a point at the end of what version it's on. And this accession number is a unique identifier for this exact gene. So if you'd like, you can copy and paste this into the search bar, and you'll get taken right back to this page. This accession number is what we use to save the sequences we'd like to come back to. So we just keep it in a spreadsheet, and we can if we want to refer back to it, we can easily just use that accession number. Next, we have the source and from Homo sapiens. So before we get to the rest of this document, we're going to look at a little bit of the biology behind this. So you can think the 9844 base pair nucleotide sequence in this case is the span from this 5' all the way to this 3', right? It's going to include these upstream enhancers, promoter sequences, tata box exons, and introns, because we're working with DNA. So these upstream enhancers help the RNA polymerase, it's called, bind to our DNA strand, and the RNA polymerase will bind right here to these promoter sequences. Now, once RNA polymerase is basically responsible for making this precursor mRNA. And once it's bound, it needs to know when to start to code for things, right? And that's where this Tata box comes in. The Tata box is basically a green light for the RNA polymerase to come in and start grabbing the information from the DNA. And now we have exons and introns. So exons, you could think of direct sequences that are used to code for amino acids, and then the introns usually just get kicked out. So right after the RNA polymerase comes through, we get into the precursor mRNA stage, right? So we have the exons and the introns, everything, and it's taken to a small organelle called the spliceosome, where the precursor mRNA is processed. And sometimes during this processing, what happens is the formation of isoforms happen, right? And that's when exons start to get mix matched. So you get very similar proteins with just a tiny difference in them, right? And now this leads us to some interesting findings. So when we started looking at a bunch of these casein genes, we started to notice, we have the Tasmanian devil here with the same alpha S1 casein gene, but there's 6 versions, right? 1 through 6, isoforms 1 through 6. So what's leading to that? And it's this phenomenon called alternative splicing that's leading to these 6 different versions. So now in this case, we're dealing with DNA, so we don't have any alternative splices, but now we can understand some of this information a bit better. So under this regulatory category, we have the regulatory class is Tata box. So that's at position 330 to 336. And then we have a bunch of exons and introns that we can look at. Now, going back to the top, we have this FASTA button, which is really important to us. So FASTA is a file type that we use to feed different bioinformatics tools information, so the sequences that we want to work with. And the FASTA file is basically a text file with a very specific format that looks just like this. So once we click that little hyperlink, we're going to be taken to a page that looks like this. It's just basically a little caret, our version number with the name of the gene, and then the entire sequence below it. Now, usually we'd want to put this into a file to do some other analyses with, but that concludes this first workshop. So now we're going to get into our casein gene cluster evolution sub-project under our Narwhal phylogenetics project. So this project, so overviewing the project, so we've noticed that some mammals have lost their alpha S1 and alpha S2 caseins. So we really want to see how these gene clusters evolved how the casein gene clusters evolved in these, especially in whales. We want to, and some of its land compatriots like hippos, and hippos are the closest land relative, which has never been done before. So we're looking to publish a paper on really looking at the differences between the casein gene clusters and all these whales. Now, this is a figure that we really want to sort of recreate. So in here we have the platypus, the possum, the cattle, the mouse, and the human casein gene clusters. And as we can see, we can get some pretty important information here, right? So here we have CSN3, which is kappa casein in all of these, the platypus, possum, cattle, mouse, and human. And from this, we know that a common ancestor for all of these, right? The platypus, possum, cattle, mouse, and human is responsible for adding this kappa casein into the genome. However, a common ancestor for the possum, cattle, mouse, and human is responsible for adding ODAM. Keep this in the back of your mind, we're going to get back to this ODAM gene towards the end of our presentation as an interesting take on TUS development for us. But this is really what we want to recreate. We want to just compare all of these casein gene clusters in whales and see how they've really changed throughout the course of evolution. So the approach we want to take is looking at a few genes. So by few, I mean exactly 4, alpha S1, alpha S2, beta and kappa caseins, compared to hundreds of species, right? We know what these casein genes do. We want to figure out how they evolve together. So that's how we're going to do that. So this is what our spreadsheet right here looks like. We just list the name of the organism along with the type of casein and their accession number so we can easily get back to it. And then this is what our FASTA file looks like. It's towards the end of it. We can see that this is what one of our entries will look like. That was generated from this spreadsheet. So if you want to look at it in a step-by-step way, our approach is going to first start by locating our casein genes on the species that we want. And then we want to find those casein gene clusters for these genes. And to find these clusters, we basically just take our casein genes and then go one gene up and one gene downstream. We're going to be doing that in just a second. And then we want to use some multiple sequence alignment tools to see how the gene clusters have changed. So just basically comparing them on top of each other, right? Sort of like that figure that I just showed all of you. And then we want to see if we can tie some major evolutionary changes in the gene cluster to actual traits that have been studied in whale evolution. So now this gets us to our second demo, which is the casein gene cluster. So we're going to be using the NCBI's genome data viewer to locate and look at the casein gene cluster of the narwhal. So if you go to our links document, we can get to the genome data viewer. So I'm just going to check this, check the chat if we have any questions. Okay, perfect. So I'm gonna give everyone just like another 30 seconds to get to that link, then we'll just get right started.
Also, just noting we're just under halfway for time, I think.
Okay, great.
Yeah, you're doing well, thank you.
Thank you. All right, then now let's get started with this demo. So to start out, we're gonna be presented with a page that looks like this. So the first thing we wanna do is select our organism. And what we can do is the search query is just gonna be narwhal. We don't need to type in the scientific name. So we hit narwhal, hit enter, and we're gonna be taken right to the narwhal page. And then we wanna search inside of our genome. And to do that, we want to run our second search query, which is kappa casein. It's gonna get us to our kappa casein gene. So, and we hit search, and then after a couple seconds, we're gonna be given a little entry that looks like this, right? CSN3 always stands for kappa casein. And then under here, it says location. So this NW and the bunch of numbers right there, it's the accession number for the chromosome, for this narwhal chromosome that contains the CSN3. And then these numbers right here, the 50 millions, are the position on the chromosome where this kappa casein is located. So we want to double click on this, not single click, double click. It's kind of weird in that sense, but we double click and we get presented with a really clustered page. So to start out, let's sort of simplify the things. And so we have, if you notice, we have a couple of these horizontal panes with some labels. So we want to keep this first one that says NCBI Monodon Monoceros, annotation release 100, May 21st, 2019. We want to keep this one. So we want to move to the second one right below and start deleting all of those pages. So if you just leave your mouse right here on that X and just keep clicking, all of the panes should just tab up and automatically delete. And then right after you've deleted those, you want to zoom out a couple times until you can see a window that looks like this. And just in case, if you accidentally delete this top one, you can just reload the page and everything is going to come back. So let's give a couple seconds. It usually takes a second to load for people. So do that a little bit, and then we'll get right into the next part. All right, so everything should be loaded for those of you following along. So as we can see, we have our CSN3 right here. So the CSN3 gene isn't in this little box right here. It's only the green part inside, right? Same thing for this CABS1 gene, the ODAM gene, CSN2, which is beta casein, right? So we have our caseins here. And once again, we have this ODAM guy that's sitting in the middle of our genes once again, which is another foreshadow to what we're going to be looking at. But this is really how we look at current genome data, right, to find our casein gene clusters. So if we were to identify our casein gene cluster in this case, we would take one gene downstream and one gene up. So in our case, we'd go CABS1, and then we'd list CSN3, ODAM, PRR27, CSN2, and then SOLT-E1, right? But yeah, that concludes that demonstration. So now for next steps in the casein gene cluster project, we want to finish consolidating our data. So right now, a lot of our data is sitting in a spreadsheet. So we want to finish consolidating that into FASTA files that can be processed by our analyzing tools. Then we want to do our analyses. So those multiple sequence alignments, and then we want to make some figures out of those so we can then really publish and look at our results. So now we're going to jump right into our test development area of study. which we're going to go back to our casein gene cluster, right? This is the casein gene cluster in the sperm whale. And as we can see, we have kappa casein right here, we have beta casein right here, and ODAM is stuck right in the middle again, once again. And ODAM is a tooth development gene. So it has to do with making our enamels and maintaining what's called our junctional epithelium, which is, you can think of it as a protective glue that keeps our teeth in our gums. Now, this is important for narwhals because the narwhal tusk is actually an overdeveloped asymmetrical tooth. So in theory, they should have two, but there's some gene that's inactivating the second one. So there have been, I think there's one fossil or reconstructed narwhal skeleton that has two tusks, which is pretty interesting. But most narwhals just have one asymmetrical tooth. And we're already doing these evolutionary genomic analyses. So why not look into this? And also, it could give us some insight into our, both of these projects could sort of help each other out, right? Because casein genes did evolve from tooth development genes. So as the approach, this is going to be basically the mirror image of our casein gene cluster project. So we're trying to determine which genes are in play, right, in creating this narwhal tusk. So instead of only looking at 4 genes and looking at a bunch of species, we're looking at 100 to 200 genes at this point in just a few species. And by few, we just mean narwhal and then its closest whale relatives to see the differences in those genes, that sort of the big differences which can tell us where the tusk is really coming from. So if we want to think of it again in a step-by-step way, we first want to identify tooth development genes, and then we want to find what are called orthologs and paralogs of those tooth development genes. So orthologs are, they're 2 genes, the same gene in 2 separate species that have the same function, which means they evolved from some ancestral parent gene, right? And then a paralog is kind of the inverse of that. So we're going to be looking at one organism where a gene duplicated itself, and then those 2 genes start to diverge due to natural selective pressure, and they start to have different functions. And we want to find these tooth development genes in some of its close relatives in the narwhal. And then after that, we want to see which tooth development genes have really diverged in the narwhal compared to the other whales, which will again tell us the genes that are really responsible for this narwhal tusk. So this gets us to our 3rd workshop, which is going to be getting some FASTA entries. So when we're working with a lot of data, so going back to this slide right here, right? 4 species, 100 to 200 genes, that's in the ballpark of 400 to 800 genes. And there's no way anyone wants to go and manually search for FASTA entries on GenBank for 400 different genes. So if we already have our accession numbers, we can actually automate the process with some Python, a package called BioPython, and just run that simple script. So we're going to be converting a list of 4 mRNA sequences in this case instead of DNA sequences, and we're going to be putting them into a format that can be copied and pasted into a FASTA file. So we're going to be using Google Colab. It's similar to Jupyter Notebook if you're familiar with that. If you're not, just think of it as an online coding environment. So first thing you want to do when you take our link, which is going to, again, on the links document, you want to copy it to your own Google Drive. So you're unfortunately not going to be able to edit this document, but once you copy it to your drive, you're going to be presented with a page like this. I'll give just a couple seconds for the people following along to catch up, and then we'll get right to it. All right, then. So first thing we want to do is install BioPython. So BioPython is a Python package that has a bunch of these bioinformatics tools built in, which make our lives really, really easy. So the first thing we want to do is on this gray area, which is a code area, we want to put our mouse over the brackets and then click that play button. And it's going to install BioPython. You should get a little console that looks just like this. And then we want to look at our list of genes that we want to test. We're going to do a phylogenetic analyses and sequence alignment, multiple sequence alignment right after this. But Here we have the beta caseins in mRNA form for 4 different organisms: the narwhal, human, horse, and southern white rhinoceros. I'm going to go into why I chose those animals in specific in just a second, but we have our accession numbers right here, right at the end. So now we have to just quickly make our script on the bottom one. So if we just click on our script down here, we have to make our sequence list, right? So the way we do that is, if you're familiar with Python, you just want to put them in quotes and separate them. If you're not, so we're going to put our accession number in quotes. So it's going to be quote, accession number, quote, and then you want to put a comma and space, and then the next one in the same format. So what that's going to look like, it's going to look exactly like this. And next, we have to set up an email. So unfortunately, you have to enter an email in order to access this database, which is, it says Entree, but it's basically the NCBI GenBank database. And unfortunately, you do have to add your email. They don't send you anything, but you can probably put a spam email in there if you really want. But once you've added your email inside the quotes, we're good to run. So I'm going to quickly explain what this little for loop is doing right here. So right here, we're basically going to be iterating through each of these accession numbers in the list. And we're going to be using the efetch function, part of the Entree package, which is basically going to fetch things from this database, right? So we're specifying the sub database first. So remember how on the first demonstration, we made sure that when we were searching GenBank, we were under that, in that nucleotide section. So this new core means nucleotide. It's just a specific name for the nucleotide section on GenBank. ID is just going to be our accession numbers, and our the ID is basically like taken from this I right here. And then return type, we want it to be in the FASTA format, and it's going to return it in a text form. And then for us, it's just going to print it. We're not putting it into any files. So once we hit the play button, we're going to be, there we go. We're going to be given a little console with all of our accession numbers. So we want to save this tab really quick. I'm just going to check the chat really quickly. It looks like we have some stuff. Oh, okay, perfect. Patrick is answering these. Perfect. So yeah, we have our FASTA entries. So we want to save this tab. We're going to come right back to it in like 2 seconds. I'm just going to go over the end of our Narwhal test development study. So our next steps would be to continue to analyze ODAM. So we've started to do some multiple sequence alignments, which have shown us some pretty interesting things. We want to continue to finish that analysis up. And then we want to start looking at the other tooth development genes that really play a role in the creation of this tusk. So now we're going to go back to our final workshop, which is going to be sequence alignment and making some phylogenetic trees. So we're going to be taking our previous 4 FASTA entries, and we're going to be making doing a multiple sequence alignment using a tool called Muscle, and then we want to make a subsequent phylogenetic tree out of that. So when we take this link, again, it's in the links document, we're going to be presented with a screen that looks just like this, right? We just paste in our FASTA entries, and we basically want to simply paste it in. You can also upload a file if you'd really like, but once we pasted it in, it should look like this. Is what the top should look like. You want to just grab the top all the way to the bottom on your Google Colab. And then we want to keep all these options as the default options, and then we want to submit. And it's going to take a couple seconds to run, but once we're finished running, we're going to be presented with a page that looks like this. I'll wait up for just a couple seconds to let people run this. All right, great. So this should be done by now for those of you following along. So right here, unfortunately, it didn't save the names, even though it's in the title for those FASTA entries, but I just wrote them out big in a big font for you all. So it's in order. So the top one's human, then narwhal, then horse, then rhino. And as we can see, it's basically putting all of these sequences in, putting their similar areas together, right? So if we look at this top one, the T and the C for the narwhal and the horse are a bit different, but then we go A, T, C, C, A, C, T, all the way to here, right? And it's the same. And then the C and the G are a bit different, and the C and the T are a bit different, but that's the basic gist of it. So next, we want to make a phylogenetic tree out of this, and it should automatically pop up. And again, it's not showing the names of the organisms, but this is what they are in order with their respective accession numbers. And we can see that the horse and the rhino, they're both what are called ungulates. So they're hoofed mammals, and they have very similar strands of mRNA. And then the human and the narwhal ones are a bit farther fetched. So this gets us into a technique called ancestral sequence reconstruction. So in this case, this is the imaginary phylogeny of unicorns, right? This is not real at all. But so this approach is being taken by many scientists, especially in recent news. It's maybe the mammoth. They're reconstructing the mammoth genome based on its relatives that they can get DNA for. And by making a tree like this, they can basically use an algorithm that'll make a sort of hybrid genome. So in this case, we'd want a hybrid genome for the narwhal, horse, and rhino. That'll be the unicorn genome, right? Because the unicorn is sort of the parent in this case, right? In this imaginary case of the horse, rhino, and narwhal lines. But we're not going to be doing too much of this ancestral sequence reconstruction, but it's being done right now with these modern bioinformatics tools and whatnot. But yeah, in conclusion, we went over our two projects. We overviewed the projects, and we did 4 different demos on different bioinformatics tools that we're currently using. And at the same time, we went over our 2 different studies on the casein gene cluster and TOS development. But once again, we'd love to have you all. Our meetings are Mondays from 7 to 9 p.m. Pacific time. We focus on real vegan cheese from 7 to 7:45, and then we start narwhal at about 7:45 and end at 9. But yeah, let's open up the floor to some questions now. So yeah, we got a little bit of time.
We have 10 minutes.
Yeah.
Well done, Sid.
Thank you.
Good work. So please ask questions, particularly if this is very new to you, and particularly if you're one of our non-binary or women folk on the channel.
So the vegan cheese project that this fits into is a long-standing community project that's run by Maria Chavez and myself out of BioCurious and Counterculture Labs. And the narwhal offspring sort of started as a lark, but we're doing some real research here that can lead to at least 2 publishable papers, and we would love to have more authors on those papers. So come join us.
Right. Yeah. And then that phylogenetic tree I was talking about earlier is a real way of thinking of it, right? This makes sense, but it is very suggestive. And who knows, maybe unicorns did exist, maybe they didn't, but.
So where it's got the little unicorn picture there, that should actually be labeled ungulates. So that is the hoofed mammals. And interestingly, whales are included in the hoofed mammals because they actually originated from species like hippos, for example, which are considered hoofed.
Right, I see.
Any questions from the peanut gallery?
What's the international term for a peanut gallery, Patrick?
I don't know.
And folks, feel free to put a question in the chat if you prefer, if you're having trouble unmuting or if you're got background noise.
So one question people often ask is, are we actually going to make narwhal cheese? And the answer is yes, we can.
Yes, we can.
You know, if we have the DNA sequence, finding the casein genes in narwhal, that's the easy part. But this was a great project to do while we were all in, you know, COVID lockdown, because it allows us to do some real science that can be done purely computationally, and is still directly related to what we're working on in this project.
Hi, I think I have a really general question.
Yeah, for sure.
But I was just curious. You said the narwhal genome was released in 2019. So,
Yeah.
like, what does it take to get a genome released, and how often are new sequence or like new species analyzed?
Yeah. Well, it's a lot of work because as you can see, going back to that narwhal page, the kappa casein part was on nucleotide 50 million on just one of the chromosomes, right? So what you have to do is get these DNA samples and just run a bunch of these sequences, right? Run it through an actual sequencer, which then you have to do a bunch of data processing, and it's a huge amount of data, a couple gigabytes at least, right? That has to be processed and then annotated in some cases to be finally uploaded. But as of frequency, I'm not too sure. I mean, it sort of depends. I mean, if there are multiple people working on different organisms and whatnot, but it depends on the different problems they're facing. So on one of the other side projects I've been involved with, the cuttlefish project, we were having a lot of trouble getting sequences for that project because we think it's the ink, their ink sac. When we were trying to take DNA samples, the ink in there will basically mess up our DNA extraction. So we'd never be able to get a solid DNA sample to then analyze. But yeah, it's very subjective on the species and whatnot.
You also have some polysaccharide issues there as well.
Yeah, that's what we thought it was.
So BioCurious has a really cool cuttlefish project that you should totally check out. If you go to their meetup page for BioCurious, you can find it on there. So sequencing a large complex eukaryote like this is very complicated. Sequencing a single bacteria is actually surprisingly easy, and it's getting more and more affordable. It's actually a really nice exercise to do for a community lab. The prices for sequencing a single bacterial genome, lowest I saw was less than $100. So large eukaryotic genomes, you know, they often require a team of multiple academic labs from around the world. In this case, narwhal, you have to go to the Arctic and actually get samples. And we were in touch with a team at UC Santa Cruz that was planning on doing some sampling expeditions for a couple of years, but some other folks beat them to the punch, I think.
Not necessarily straightforward to go and take narwhal samples. So folks, we've got about 4 minutes left, so keep the questions coming. Thank you, Anne.
Well, if there's no more questions, we might end a little bit early. Now we'll prepare for the next talk.
All right then, perfect.
Thank you, everybody. Please add yourself to the notes if you haven't already, and I'll re-pop them in. And yeah, please join us for the next workshop, which is kombucha leather, I believe.
Yes. All right, thank you all. So if you are one of the organizers, please stay around. If you are not one of the organizing team, please leave the Zoom room so we have some chance to set up for the next one. And thank you all for joining, and well, maybe we'll see you back in 15 minutes.